Can you see my browser with the day 3 classification? All right.
Yes.
Yes.
Thank you. So we are trying to find in the first step the closest relatives for our phage. So we have 2 phages that we isolate, well, that we sequenced. We have the genome, we have the correctly oriented genome, and we have the annotations. So what we're going to do now is use NCBI Virus to find the database relatives. I am going to use T7 as the example for the genomic bit. So oh no, it's not in here. I need to open it from. So what I'm doing off screen is just opening the text file of T7, doing Command A, Command copy for myself, and I'm going to go to NCBI Virus, and I'm going to do search by sequence. So I'm just going to paste in the whole FASTA formatted genome of T7 that we restarted, and I'm going to do start the search. So this is a relatively new part of NCBI that is specifically developed for virus, including phage people. If we look at the page and look at it in a little bit more detail, so now we did the nucleotide search with the whole genome. Obviously, you can't do that for a bacterial genome, but phage genomes, it works quite well. You could also do a protein search, obviously, and even the example is of T4. It is of a protein of T4, which is quite funny. But you can also search by virus name or taxonomy. And when you start typing here, it will start making suggestions for certain groupings. Or you can just click here and search for all bacteriophages, for example. Let's see whether, oh no, the Zoom is overlaying my my browser. Okay, we have a result. So what I can see here now is that it's going to be limited to 499, I think, but we have 499 records. I don't know exactly what the original sorting algorithm is, but it's a bit all over the place because the values that we look for are coverage, percentage coverage, and percentage identity, meaning my phage T7 restart aligns with this one for 40% of the genome, and the percentage identity is 77%. Anna, do you have a question now?
Yeah, sorry, I was mute. I wanted to know if you don't have the restart knowledge, if you're working with something that you don't know exactly where it will restart it. Oh. So your result here, this is gonna affect a lot or not?
For blast-based searches, it's not really going to make a difference because it automatically takes that into account. But it will, for example, in the output, kind of say that, you know, the bit at the front and the bit of at the back are now similar, and both are similar at 100% or something like that. But whereas if you restart it at the correct spot, it will just say 100% similar from base 1 to base the end. But the percentages will usually match, if you understand what I'm saying.
Yeah.
Okay, so what I like about this NCBI virus thing is that it's very easy to refine your results and refine what you see. So if I want to know what the ones are that are most similar, I'm just going to press click on the coverage. Oh, I clicked on the correct button. So now I can see my table is rearranged, and I can see the ones that are the closest related first, which is good because I'm actually looking for my most closely related phages. So now I can already see 100% coverage, 99% identity. My phage is not a new, does not represent a new species. Maybe you find this a bit weird with the accession and the submitters and the release date. You can choose select columns, and I'm like, I don't want to know who the submitters are. I don't want to see the release date. I don't want to see the isolate, but I want to see the genus and the family, for example. And I want to have the GenBank title, apply. So now I can see that, you know, all of these are assigned to the species Escherichia virus T7. That makes sense. In the genus T. septima virus, in the family Autographiviridae. Excellent. Host E. coli, GenBank title. Here you can see the actual name of the phage or of the submission. Sorry, I'm opening the chat. So now I can also see what you guys say there. Okay, so now we have, you know, our T7 genome that we submitted, and we want to download our closest relatives, but 499 other genomes to download is a little bit much. So we can use the left-hand side here to refine. So we can, for example, in sequence type, check, oh, we only want the RefSeq sequences. And then we're down to 72, meaning that we should, if all goes well, only have one representative per species here. We can also refine for completeness. Let's just limit ourselves to one more. So now we're all complete. So there's loads of things. So if you have, for phages, it's usually not that much of an issue, but if you're, for example, dealing with another virus that is very geographically distinct, then you can, you know, search for geographic region, for example. Anna, do you have another question?
Yes. So what will be your cutoff for the coverage and for identity to trust that this is the species that you have identified?
So the first thing we're going to do is we're going to select a bit of a range of things to further investigate. So I don't want to just, so in this case, because it's 100% identical over 99% of the length, we know that it's, you know, Escherichia virus T7, which will be renamed to a binomial format in the coming year. So for in this case, you basically follow everything for this species. But I will show you how you can assess the species and the genus boundaries from data that you've selected from here. So I'm just going to select a number of these, but I don't want to overload my following tools. So I can see that I've selected 9. I'm going to select, let's say, 12 randomly from here. And then I'm going to scroll a little bit further down, and I'm going to select 2 more from, let's do an Enterobacter. And if I switch this one off, I am going to, oops, I can't click too fast. I'm going to switch this one off as well. Oh, I've reset. Let's start again. So I'm going to first select a couple of the similar ones. Okay, 5 similar ones. And I'll refine with some of the, why do you always reset? Okay. Let's just do it like this. I don't want 72 because it will overload my program, my next program. If you refine your search to a manageable number that's here, you can use this button to select them all, but I don't want that many. Okay, 18. Gonna do download, nucleotide, download only the selected ones. Next, I'm just going to use their default settings for what's in the FASTA definition line, and it will start downloading. And then I will have my sequences.fasta. And so if I now want to compare them, I'm going to, you won't be able to see that now, I'm guessing. I'm going to combine this with my own phage genome into one FASTA file. I will save. Okay. So what the next thing that we're going to do, Hani?
Yeah, what about the tool available actually at the virus website about the build the phylogenetic tree and online?
Okay. Yes, we can have a look at this. But I was trying it out earlier in the week, and I had to actually log a ticket with the people who built this website, because it says that you can build from up to 500 sequences, but it's not actually the case. And the person who was behind the help ticket said, "Oh, you'll have to, start increasing the number. Try with 5 if that works. Try with 10." So, and then I didn't really go much further. Well, we can see what it does if it works for 72. Okay, so it's building multiple alignment. That's something. And I'm going to try, let's see. But this is something, I mean, this is online. You can play around with this yourself. This is relatively new for me as well. So I'm not super familiar with it. Seeing, okay, this gives a loading error. This is most likely just because I've selected too many. Let's see, the multiple alignment is still going. So remember what Andreas said earlier about phylogenetic trees and multiple sequence alignment. In this case, it says build a phylogenetic tree, but it's not actually based on the multiple sequence alignment. It's based on the distance results of the BLAST search. And the BLAST search is a pairwise search. So it's not a phylogenetic tree like we will learn how to make one later today. See what it is doing. This one. Okay, so if I selected only a couple of them, you can see kind of a tree in internal program called Genome Workbench, which is an internal program from NCBI that you can download to your own computer and also work with. I find it sometimes horribly complicated. So in this case, you can see the search results, but what you can't see is actually your own sequence in this tree, because it will show you the tree of only the search results. And this one is still going. So while we wait for this one, let's go to Veridic. So Veridic is a very relatively simple tool. It's developed by a collaborator, Christina Moraru, in collaboration with Andrew. And it's basically to generate sort of an overview of the intergenomic distances or the nucleotide similarities of a bunch of bacteriophages. So what I'm going to do is I'm going to make a project, the username and a project, and what we're gonna do is T7 likes. I've probably already tried this. I am, let's see whether this works. I saved it in my downloads actually. Sequence. We're going, so one of the things that it's based on, it's based on pairwise comparison of each genome to each other using BLAST-N, and it gives you thresholds that you can change yourself, but it's preset for 95% and 70%. And let's see, if you press create project, then it should so it said that my FASTA file was valid, which is something. So that means I can now run the project, and now it's going to be calculating, and I need to leave it alone for a while. Let's check back here. This one is still processing as well. So we are in a bit of a waiting game at the moment. Meanwhile, I'll show you that you can just play around with this as well, because you can click around on some of the viral taxa. If you're interested in DNA viruses, then you can see the type of families that are currently defined straight in here, or the type of realms that are present with the DNA viruses. For example, what I'm interested in is DNA viria, and I'm interested in the Urovericota, and I'm interested in the Caudiralis. And I can see that, there's lots here, lots of different subfamilies, families, and things defined. And we can see the host distribution, and it's mostly what used to be Mycobacterium smegmatis, and that's because of the Phage Hunters program. We use that bacterium as their host, and then E. coli and Salmonella, then another C. phages host, and so forth. Anna, do you have a question?
Yes. So I want to ask if you could show how the file containing all the sequence looks like, because it did it like in the hide mode.
Yes. Let me see if I can share, new share. Okay, this one. Okay, so this is what I downloaded. So can you see this text file?
Yeah, I can. Thank you.
So,
yeah, that's what I wanted to know. If
yeah.
they were all together on the same, having the very simplified FASTA.
So this is basically what I did. Is the first sequence I pasted in, which was my T7 restart.
Okay.
And then the rest of it was the automatic download, and I didn't change anything. It just came like this.
All the downloads were together, and then you just added
Yes.
the beginning, the other one. Perfect.
Yes, they were all in the same file. So if you go further, you can see all in the same file.
Thank you very much.
So that's the upside for me about the NCBI virus. It easily allows you to download all of them into one thing that you then use further. This one is still calculating. This one is also still calculating. So so then we'll just have to wait. Yes, Andrea said from the command line you can concatenate the files. I've also written this somewhere here. Okay, so this is a simple one. Let's say the file that I download, I named t7likes.fasta, and my own genome was t7restart.fasta. I concatenate this, and then I name the new one T7 for Viridic FASTA, for example, or combined.fasta or something like that. So you can easily do that, but in this case, because the file's not that big and the genomes are not that big, it's easily opened. If you deal with files that are thousands of or genomes long, and your own genome is very long, then it gets prohibitively more computationally expensive to open these type of files. Okay, in the meantime, Viridic has finished running. So it will show you a heat map with intergenomic similarities, and it has a color scheme and things that it chooses as default settings, and all of these type of things for the heat map you can change here in this setting and run for yourself. And it also comes with a table that you can download, but it shows you the first 10 entries here, where you can see we're comparing, for example, genome 0477 with our T7 restart in this location. And then it will suggest, based on the clustering, new species and new genera. And you can also download these clusters. Let's look at the heat map. Can you see this? So we already know that we downloaded stuff that was genomically similar, right? So in this case, that means on our top right diagonal, we always have shared percentages of intergenomic similarity. But let's go through this output in a little bit more detail. So you see 3 color codes here, and you see a kind of a bar chart here. So the bar chart is for each of the genomes, which are in each of the columns and each of the rows, its genome length. So all of them are around 40,000, except for this one. This is slightly shorter. It might be because it's partial, or because it might be being logged as complete genome, but it isn't actually a complete genome. Then it will give you the aligned genome fraction at the bottom left diagonal and the genome length ratio also at the bottom left. So basically, what we're looking for is a more white color, because we want to have all of the genome aligned, so a value of 1, and the genome length ratio also to be close to 1, meaning that the 2 genomes that are compared are the same size and the alignment is over the whole length of the genome. So we can look at, for example, here for the combination of the first and the second genome, it's actually only aligned over 70% of the genome fraction, but they're the same size because this is 1. What we're most interested in this part is basically our intergenomic distance between the 2. So when I look at the T7 restart, which is here, then I can see that it's 100% identical with this one. And I hope that this is the reference genome for T7. That would be great. But then we can see with this this section, they're all more than 80%, but so definitely more than 70% similar between their whole genomes. So these represent a genus together, and this should be the T. septimovirus genus, whereas these also belong together. And we can go back to the NCBI virus to see whether they were indeed a new genus or whether I have some work to do. Just gonna do and look at this. This is actually a T3 virus. So different genus, great. So and then we can see that these 2 are again more by themselves, but all of these are still genomically related, but not in at the species or the genus level together. So this is where in the realm of these, if we go below 70 and definitely below 50% intergenomic similarity, that's where we need to look at the protein level. And if you want to kind of redefine the whole lot, you can do that, but not just at the genomic similarity level. Is everybody with me when it comes to interpreting a plot like this? Okay, I can see 2 thumbs up. I'm gonna take this as a go ahead. So this is, if your genome falls somewhere within this, and you can see that either the species it belongs to or the genus it belongs to has been defined already, then there's really nothing more you need to do. You have now all of the information to fill in your GenBank file. But you might want to create a nice phylogenetic tree. Oh, Melinda?
Is this something, if you're describing your bacteriophage and publishing it, is this something that you should put in the paper, or is it just for information?
I would always just put that in as one line.
So not the picture?
No, no, unless what you're doing is reassessing, the genus and finding other members that are unclassified in the databases and you want to describe that. If it's just about your genome and it is in a genus that is well defined, in a species that is well defined, I would just say that in one line and you can already see that from the sequencing, from the main BLAST result. If you have 100% 99% together, then you can just say phage Melinda 1 belongs to the species Escherichia virus D7, in the gene is the Septima virus, and leave it at that, and then in your paper talk about your phage rather than about the taxonomy.
I really wish they'd let me call my phages Melinda 1.
They should.
I mean, there's nobody stopping you but your boss. I will not stop here. Okay, so we have now seen how we assess basically taxonomy at the nucleotide level. Let's go back to NCBI virus and let's go from the hypothetical situation that you don't find anything when you do your genome searches, and you go to VipTree. And I submitted RT4 to VipTree before, and which is available from this address, and it's also here in the somewhere using VipTree. So if you click on the link, you can also find it. So I submitted T4 as the whole genome, and I chose double-stranded DNA as the database and prokaryotic as the host, and then I just submitted. So I'm going to start to browse the results. So you can see that I wasn't lying. And now I can look at the related genomes and look at the tree. So I'm going to click on circular tree. So now it's kind of loading everything. And now it gives us a subset of the tree with stuff that is related, and my phage is located here, and this whole bunch of things is going to be all T4-like phages. One of the things that you can see here and that you need to be careful for is that VipTree does this logarithmic scaling of its branches by default. So you can see that this bit from here to here is only 0.01 in length. So if you want to scale that to linear and redraw the tree, you can see it on the same scale as the tree I showed during my presentation. And then you can see what happens. So for example, if I wanted to do a reassessment of the whole kind of group where my phage would be similar to, I can do either choose this whole clade or go further and choose like something like whole of this, or even further. Not, once you reach here, then there's no shared similarity anymore. But so this would be a way for you to identify non-genomic relatives if your phage were to fall in a very deep line. Okay, this is just as an illustration. I am not going to now take this any further. So if you have any questions about VipTree, now is the time.
Probably I would like to, so you were mentioning that Myoviridae is gonna be going to another naming. So in this case, we have the inner ring in this
Yes.
apple green color, which is, according to the legend, going to Myoviridae. So if you're using this technique to identify where is falling your sequence virus, then how you will do the classification in all these things now are being renamed.
Okay, yeah, so as you can see, this apple green, as you said it, it's here, but it's also on this side and it's also on this side, and those two share nothing. So you can't use, that's what I was illustrating, saying you can't use Myoviridae alone, and that's why we're getting rid of it. So now these ones will be, in the next taxonomy release, will be two new families at least, like I showed you in the presentation. But there are many ways to investigate this tree with data from within here. So any one of the taxa, the leaves of the tree, are present here with information. So you can use that information to then download a set of genomes and start from there.
Okay, so basically, if my heat here is now matching from that upper table the option 157, then I copy that 157 option and I blast it apart to see what more information I got about it.
Yeah, so basically what you do is you kind of take note of, you don't necessarily have to download the whole genome, but you have to know, you have to have a look whether, you know, there's other families as well. For example, here there's Ackermann Viridae, which are here now as others. So, you know, you can select a bunch of these, not necessarily on this tree, but you can investigate the rectangular tree. Let's just do that and have a little bit more detail. So if you look at the rectangular tree, you can, okay, have a look. Mine is now here. And then you can have a look at all of the other phages that are in the clade. So you have more information, and you can download this, and you can download the table, and you can look at, you can select some of these and browse the alignments here. So you can have a whole bunch of, you know, you can investigate a lot of things within this browser system before you necessarily go on to the next stage. And for me, the next, so it's always about identifying a set of related genomes to then go more into detail with investigations.
Okay, thanks.
Okay, so for yourself, like, you can play around with this as much as you'd like. What were we saying? So we did first finding closest genomic relatives of your phage, actually, and then assessing the intergenomic distances. Then we are going to, I should have, we'll come back to point 3 in a bit, but first we're going to go point 4 and select conserved genes to build a marker gene tree. So like I said before, some of these, you can do a core genome analysis with, you know, all members of the family, for example, to identify all of the genes that are shared between all of the members. Or you can use information from previous publications to see what is a good marker gene for this set of phages that are related to mine. So what I'm going to do now is I'm going to back to what we did yesterday, and I'm going back to my T4 genome. And I'm going to search for the terminase gene. And now it's going to work, I'm sure of it. Why aren't you doing it? I swear to you guys, I tried this before and then it worked. find qualifier. Did you now find any? This is always the case when you try something beforehand, then doesn't want to do it while you're all watching. Sigh. Alternatively, I'm quickly going to do the same with Control F in my text GenBank file and find it from there and quickly copy it, which I'm doing behind the scenes right now. So if ever you get better at Eugene than I am, this will be much easier for you. So I have in the background now copied the found determinase large subunit in my file. And I am going to do a search by sequence protein search here. Start search for all of our terminase genes to see what that brings us. Has the multiple sequence alignment? No, again, honey, I'm sorry. Too many sequences and I didn't want to do anything. And it timed the request timed out. So I'm going to close these things.
Yeah, I just wanted you to try it because it didn't work.
Yeah, absolutely.
Yes, to tell them.
So yeah. So if you
and again, could you please highlight again the difference between the phylogenetic tree that can be produced by the virus and what we are going to do later, or what actually Andrea explained? Is there any difference between the phylogenetic tree that we can produce with the NCBI tool?
Yeah. So here the tree is actually a tree that is based on your BLAST results. So BLAST results are results that are pairwise with your search query. Although, let me think about it, because it might be a proper phylogenetic tree, but the problem to use that is that your own search is not included in the trees and in the alignments. It's only the things you select here in your search results. So it's interesting to have a little view of these things, but because it doesn't include your own sequence, it's not always very useful for your own publications.
Exactly.
So, but there's a button here where you can view the BLAST alignment of your sequences, and then you get a graphical view of, again, in this program called Genome Workbench, and then you can have a better look at in detail at the exact alignments and the exact search results. And I'm very new to this kind of investigation of this program, of alignments with this program as well. So it gives you a lot of information. If you click on something, you can investigate every single base, and so it's showing us now only a couple of BLAST results for a couple of phages, but you can investigate this further if you are interested in this. Okay, we pasted that, we did start. Okay, so I found 491 terminase proteins. My own protein was 610 amino acids long. It used the search algorithm BLAST-P, and it looked in the viral protein collection of NCBI. I'm going to have a look and see if I can select my columns. I want to look at some coverage and identity scores. I don't care about the release date. I don't care about the isolate. Maybe I want some genus information as well. And I need to press apply. So now you can see here the coverage and identity scores of my BLAST results again, and you have information on what species the phage belongs to which this protein belongs, and the genus. So I can again sort by coverage. And then we can see that there are a couple of them that are very, very similar, which is normal because we've sequenced T4. We can limit ourselves again to the RefSeq database. So then we have 133. For example, and we can see that all of them are very similar. So when I ran this the first time, I'm going to be honest with you, I made a little mistake and I only downloaded the ones that were super similar. And then eventually your tree will be slightly ridiculous because everything will be the same. So I'm going to try and not make that mistake this time. And I will download two subsets, a subset that is very similar and a subset that can be counted as outliers. So I'm going to choose the very similar ones first. I'm going to choose, let's say, 7. I'm going to choose a couple of more from more down below here. You, and you. So I can see I have 11 now. I'm going to download them.
Excuse me, can I ask a question?
Yes.
What would you say would be a good practice when selecting these proteins? Like, for example, let's say if I keep scrolling down until I find one that has 50%, should I select that one? Would that be not recommended? Because I mean, it depends on the selection criteria that like everyone has, right? So
yeah. So one of the things is that when you build a tree is that you can't have too many sequences, and then it depends also on the gene length. So how many amino acids do you have? Luckily, we have a large gene, so we have a lot of leeway to select a lot of sequences, but the more sequences you have, the longer the multiple sequence alignment takes and the longer the tree building takes and the more complicated it becomes to visualize it. So it's a matter of how many relatives are available from the database and your kind of computational power, I would say. So I'm going to show you now, because I'm going to combine kind of two search results into one tree, one for the similar ones, which I found using the BLAST, and ones for more dissimilar ones that I will use a search term for. So I'm in the process of downloading my set of 11 from the beginning. I'm just going to do a little bit more, actually. Sorry. I'm going to go up to 20. I'm just randomly adding some. 20. Okay. It's protein, selected records, next. I'm going to download this. And not simultaneously, now I'm also, I know I sequenced T4, and I know, or I've seen based on my searches here that my closest relatives are T4 virus and mosaic virus and DACA virus and Karen virus. So now you can use that information to look where that's situated in the taxonomy tree. Now I know, because I know where they are, I know that they're in the subfamily Tivenverinae. So I'm gonna look for those. Tivenverinae. I'm going to click on it. So now the search results will show me all T-VIN VRNA, nucleotide, protein, and RefSeq genomes. Now I can use the refined search function for proteins to again search for my terminase, and I'm going to look for my terminase large subunit. So I'm going to only look for the ones where it's properly annotated, because you can see some people do terminase large subunit, some people do large terminase protein, some people do terminase family protein, and again, this is where high quality annotations come into play. So I'm going to look for these. This should limit my query again. And I'm going to select my columns. What do I want to show? Genomes as well. Apply. So now I want to no isolate your. So now I'm going to look for some of the genera that I maybe didn't have yet. Or something that doesn't have a genus sounds nice. This one, this one, this one, you, and you can also join. Or maybe I already have you, we'll see. Okay. I'll do a sudo C even and I'll skip to Shizu T4 viruses as well. Okay, now I've made a list of 10. I'm gonna download these as well. Protein, selected records, download. So now I'm going to show you how you do your phylogenetics. And in my case, this is a lot of, this is also, not, I'm not going to say a lot of trial and error, but this is a bit of a trial and error, especially when you don't know your phage and your sequence space, taxonomic space around your phage well. It's a bit of a trial and error to download the correct ones with the correct outliers to get a tree that is reproducible. So the ones that I've downloaded, now when I download it all comes as sequences.fasta, so I'm quickly going to have to rename these behind the scenes. I'm going to rename them T4 1 and T42, and then we are moving on to our command line portion of the day. So I'm going to use CyberDuck to I have made a. Are you seeing my CyberDuck or not? Okay, I made a folder.
Yes.
I made a folder called tree building. There's already one file in it, but that's the file that I made with the sequences that were super similar. I'm dragging my other ones in there. And I am also going to put my t4.gbk in there. Just to have it, and what I also need is the original file of the terminase of my t4.gbk. Oh yeah, I couldn't download it couldn't find it again with this because Eugene doesn't like me at all.
Can I quickly ask another question about the selection of the marker?
Yes.
You said you mentioned it depends on the phage, right? So you recommend going to the ICTB website and look at the taxonomy proposal for that specific phage and see what marker was used, right?
For that particular group of phages, if you know in what broadly what group it is, yes.
I see. Okay, so I'm assuming all the proposals have the same format where you can easily identify which marker was used. Right?
Well, all of the if you look at the proposals, most of them, except for if it's the really small ones, but the larger ones should have a phylogenetic, at least one phylogenetic tree in it, and in the legend it should state which marker gene was used. If it's one of the new families, the publications or the ICTP reports usually have more information on this as well. If there's no information, then a good place to start is a terminase large subunit, the capsid protein, or or a polymerase. So not all phages encode a polymerase, but when they do, those are good ones, because then you automatically, are already combining phages that are more or less related because they all share a similar polymerase.
Okay, thanks a lot.
What did sorry, I lost my train of thought for a second there. I'm quickly also going to make a file with the amino acid sequence of my own terminase so that I can combine it all. And you can probably hear me typing. So I'm also uploading this if I remember where I saved it, which is always a mission. Oh, never mind. Okay. Back to our terminal. I hope I remember what my login information was. And remember how to type. Excellent. I'm going to quickly check whether what I copied is actually available. Tree building. Okay. I'm going to, I have a very awful way of making new files that bioinformaticians may not like.
It's the same I use.
Okay. But now.
Command plus a bit to increase the font size.
Oh yeah, sorry. Better? Okay. No. What did I do now? Now you can see me faffing about on the command line because I need to copy what I wanted. Okay, so now I have my files. Normally, I'm going to move my T4 into my tree building one. So I'm going to list CD in my tree building. Okay, so now I have my terminase sequence of my T4QIB. I have my 2 files that I've downloaded, and I'm going to put them all together into one file, T4QIB, T41, T42, and I'm going to clear L fours. Okay, I should now have a file called TRL for Fasta. Excellent. The first thing that we will do is multiple sequence alignment. We have made a new environment. I say we, but it's Andrea who did it, made a new environment so that we can do some tree building on our command line. So the first thing is to check that you've cloned the repository from day 1 so that you should have a file called Phage Annotation Workshop. So I'm going to do this with you. I'm going to list my folders. Yes, there is a Phage Annotation Workshop. I'm going to change into that folder. And then you need to make sure that you have the repository cloned. If you don't find it, you need to clone the repository. That will make this folder. Because we have this folder, we don't need to do this step. We need to update it by typing git pull. And now it is updated. And then we need to create the environment. So I already did this earlier. So I'm not going to do it now because it will create some problems, I think, but I'm just going to activate it. Conda activate taxon. Okay. We can see that I'm now in my environment taxon. What we're going to use is the first tool for the multiple sequence alignment that we're going to use is MAFT. There are many multiple sequence alignment tools. Some are super famous, like Clustal W, which I think not many people still use. And you have Clustal Omega, you have Muscle. In this case, I like MAFT because it's very simple and useful for phages. So I'm just going to type MAFT just to show. Oh no, why does this not work now? I'm going to see MAFT help. Okay. In this case, it doesn't recognize the -h. You probably need to use the -- help, but you can see that it is installed and it gives some suggestions on how to use it. And in its simplest version, you just type MAFT, give your input file, > sign to assign it to a new output file. Super easy. Well, another thing I find super easy is if you're unsure about the options to use, and it has some options, you just choose auto. And that's exactly what we're going to do. I'm, as said here, I'm going to, sorry, I'm in the Phage Annotation Workshop. I'm going to go out of there into my tree building folder. Where I have the files that I need. So I'm going to type MAFT --auto, and I'm going to present it with the file for lt4s.fasta, and I'm going to have an output file that I'm just going to call MAFT for lt4. So for MAFT, you don't really need to specify an output format. So I'm not going to, and then it's going to run in the background. and that's it, because we didn't have a lot of sequences and they're all very similar. It doesn't have pose too many troubles. So you could have a look at it. Let's just look at the first 10 lines. And it's good. You can see that it inserted some gaps. So we have a multiple sequence alignment FASTA file, and I can see that because, you know, it introduced these gaps. Okay, we can use Eugene to have a better look at it, just to show what the default settings give you without any further refinement. So I'm gonna go back to CyberDuck, and I am going to download this to my, where are you, mapped. And I'm now going to open this into Eugene, mapped, open, and this is what it looks like. And this standard, it's zoomed out to show you the whole area, and it will show you your input name, the FASTA headers on the left-hand side. It will show you different colors for your amino acids, and it will show you here the positions in each column that, like in this bar chart, how many, you know, percentage-wise, how many positions are covered. So you want to have something quite high. So you can see that there are potentially some issues here with this genome, this gene, and this gene potentially being partial. So one of the decisions you could make is to just leave it out. Or you can just, you can leave it in and see, you know, it will show you as, it will probably show you as an outlier on the tree. I quickly want to run something again with the file that I had earlier. So if you remember, I had another file that was called LT4 likes. I'm gonna run math on that as well, because this one had a couple of interesting features in the multiple sequence alignment. So LT4 likes FASTA. I'm gonna do test, and it should run quite quickly as well, but this one had more. This one had 90 files, 90 sequences. Okay, I'm gonna download that one as well. Test. And I'm gonna open test in Eugene. Okay. And for this one, I just, what I did is I did exactly the same thing. I took the terminase large subunit of T4, of the QIB T4 that I had, and I did a blast, and I found all of the T4 viruses, and then I just indiscriminately selected all, downloaded all. And then I ran my multiple sequence alignment, and what do I see? Oh, there's like this whole gap of nothing being aligned except for this bit, but that's T6. But then I have T6 again. And then I have SG1 here and twice, but with different, you know, alignments. I have some T6 here again. So one of the things that's possible, and I'm going to say likely in this case without investigating it any further, is that there was an intron in the T4, or there are introns in the T4, in the T6, and those terminase large subunits, and it is present as different fragments in the database. And because I didn't specify that I only wanted fragments that were complete, in my automatic download, it now downloaded everything. And this is what happens in the multiple sequence alignment. So this is why it's important to both be mindful of which proteins and which genomes you download, and not just click the download all button, and looking at your multiple sequence alignment. Because now what you could do in Eugene is just select this and press delete. Select and press delete, saying, okay, this is going to be useless because, you know, you're T6, I don't trust you anymore. There's one T6 one that it aligns perfectly. So again, this is probably because I did the a keyword search. So for example, now it looks a lot better. So I'm not going to work with this one anymore. This is my test, but this is just to show what you can, what visualizing your multiple sequence alignment is for. I can also zoom in, and then you can, you know, really look at it more in detail of what's going on. For now, I'm just going to leave it like it is. The next step you can do is trimming the alignment. Basically, with trimming, you will remove those columns in your alignment that are too gapped. It is not a necessity, and it will depend on the type of data you give it. Because sometimes trimming steps can remove so much that you're left with almost nothing. So let's just give it a try. And I'm going to use the program called Trim All, and I'm going to use an automated feature called Gappy Out. So we want to get rid of the gaps. Trim All. I'm just going to check whether it exists. Okay, I see a help file, so it is properly installed. Trim All, Gappy Out. What's the syntax again? Dash in, dash out. Okay, so Gappy Out, Trim All will automatically recognize the type of input file and give you the output file of the same type. So we have a multiple multifasta for our multiple alignments, so it will automatically return the same type of file. So I'm not going to specify any output format again. Maft. And I'm going to rename it something like trim maft. So I always end up with progressively longer names because I want to, when I look at my files, I want to know what I've done with them. And this runs really quickly because it's already done. Again, we can have a look at it. I'm going to go back to CyberDuck and refresh, and it is here, and I'm going to download it, and I'm going to open it in my Eugene. Trim maft. And if we zoom out now, we can see that it trimmed some of those away, some of those columns in the alignment that were very sparse. You can give it settings where you say, I only want to trim away columns that have less than this much %, I think, and stuff. At the moment, we're just going to keep with the standard automated settings, the default settings. So now we have this one, and I'm going to use this going forward. So the file was named, not clear, was called Trim All. I'm just going to show you which files we had. So trim maft L T4. That's what we're going to use to build our tree. So the suite for tree building we're going to use is IQ-Tree. Oh, I can see that I did not close my brackets correctly when I was typing this. So the hyperlink is broken. Okay, so IQ-Tree is a suite of tools, actually. And they each have their own reference, meaning that when you use IQ-Tree, you should cite a bunch of papers together. And the command that I'm going to use is IQ-Tree, the input file, I'm going to use -m mfp, And that's, so earlier we talked about substitutions and evolutionary models. So evolutionary models, so this is a substitution model. So this step will ask IQ-tree to perform a model finding search that will then use the best fitting model immediately in your phylogenetic tree building. And this is just automated, and that means you don't have to think about it because it will do it for you. You just ask, please do this for me, and then your output tree will be made. So we will also ask it to run 1,000 ultra-fast bootstraps to assess branch support, and also perform the approximate likelihood ratio test to assess branch support. And we will let the system decide how many threads to use. So now I hope this is going to work. So we're going to do IQ-tree. Yes. This is our multiple sequence alignment. M, N, F, B. 1,000 ultra-fast bootstraps. That's okay. I realized I didn't try this out after typing this. And T-auto, let's see what it gives. Okay, so now it's going to run. This has a lot of underlying science in which I'm not necessarily a specialist. So if you have any questions about this, please ask them, but I cannot guarantee that I can give you the best answer for this very specialist subject matter in phylogenetic tree building while we wait. Or maybe you are all specialists in phylogenetic trees and I'm just kind of preaching to the choir. I guess from Hani's reaction, it's not the case. If you are interested, there are entire books that have been written on the subject. So if you were to not maybe want to go through all of this effort to do it on the command line, and you don't have a lot of data, one of Andrew's favorite programs is phylogeny.fr, which is a website that runs maximum likelihood trees in one-click mode. So should have opened it in a new tab. So you can see that it uses alignments using the program Muscle. We've just used MAFT, but Muscle is very good as well. It does the curation that we did with trim all with G blocks. You can switch that off here. Then it makes a phylogenetic tree with PhyML. We are using so it's a maximum likelihood tree with the program PhyML. We are using maximum likelihood tree as well, but with the IQ-tree suite. And then it has this tree visualization software built in with TreeDen. Give it a go if you like. We're not going to do that here because we're doing exactly the same thing on our command line. Okay, and it is now done, and it only took us 20 seconds. Of wall time, wall clock time. Let's see what happened in terms of the files that were created. So IQ-tree creates quite a couple of different files. And the one that I suggest you open first is the one that ends in .iq-tree. So we can use the less command to have a look at it. Tree. So it automatically takes the name of the multiple sequence alignment file and just, you know, makes its own file extensions for that. So this one kind of shows what happened. And apparently we're using the beta COVID edition. So the type of analysis that was done was we found the model for our data, we did tree reconstruction, and we did ultra-fast bootstrapping of our tree. It gives you what to cite, that is very important. It gives some information about our sequence alignment. So we had 31 sequences with 574 amino acid sites. There were 122 sites that were constant, so the same everywhere. Based on that, our model of substitution was found, and it is LG plus G4. This is the Legasquel model, which is, I think, ironically, the same person who made phylogeny.fr. So these are all the models that were tested. And you can, if you are interested, you can Google every single one of them to see what the difference between them is. So these are all models for amino acid substitution predictions and evolutionary distances. So the decision was made for this based on the Bayesian information criterion. And that is basically that compares log likelihoods. Okay, so then we're getting to our maximum likelihood tree. We can see the log likelihood of our tree. And then it will give us a little bit of a shape of our tree already, so we can have a preview of, you know, what our tree looks like from our command line. I can see that Hani trusts the automated model selection. I absolutely also trust the automated model selection, and I'm very happy with this, and do not want to question this any further. So here you can see what the tree looks like. Andrea showed you what the Newig format looks like, so that's also present here. And then there's the consensus tree. So the consensus tree is the tree that is constructed from 1,000 ultra-fast bootstrap trees. So basically what is done, we'll have to look into the paper on the ultra-fast bootstraps, but bootstrapping is basically a method where you take a subset of the columns of your tree and then remake your tree again and again, in this case, 1,000 times, and then you compare that one with each other and with your original tree, and then you use that number to create bootstrap values as branch support values. Andrea, am I telling this correctly?
Yes, this is the part of the refinement that I briefly mentioned before. So after you make a tree, the question is how strong. Of course, you have a result, but how strong it is and how well supported by your data is.
Yeah.
Not every bit that was used is as informative as the other. So more or less, this is the process that was done here.
Okay, we have reached the end of our IQ tree file. So the consensus tree is now present as the tree that ends with .con tree. And the original maximum likelihood tree is present as the tree file. So what we can do, so that's free for everybody to view in their favorite tree editor or whatever. I am a person who uses ITOL, but ITOL has become a paying software for if you want to save your stuff. But I can have a little preview of what it looks like if I download the tree, and then I can show you what it looks like. So we're going to have a look at the consensus tree, and I'm going to open ITOL in a second. I already did one. So I am in my own test tree workspace for the Phage annotation workshop. I am going to quickly show this tree. And this is what my tree looks like. So this is now just first upload of my tree as it was. I am going to do a midpoint routing just to clean it up a little bit. So now this is a way to kind of, without knowing where your root lies, to just provide a root based on the topology of the tree. So where has my own phage gone? It is now here in this cluster. So now we can use the bootstrap values, which we can display as text to see how strongly supported that each of the clusters and clades are. Now, this is an ultra-fast bootstrap, not a normal bootstrap. So one of the things that you need to take in mind is when you normally do a tree with other algorithms, you are happy with bootstrap values over 70, more or less. Ultra-fast bootstraps, you're only happy with values over 95, as per the guidance of the people who wrote the program. So, but what I can see is that this kind of cluster has a bootstrap of 100. So it's 100% of the times this clade was recreated. So I'm very happy with that. And I'm pretty sure that if I'm going to look these up, this is probably going to be the genus T4 virus. So now there's ways of overlaying information on this tree from text files to other metadata, but that's a whole another it would be a whole nother session to just do how to make my tree pretty with metadata that we're not going to go into. But if you were to, have a phage that is similar to other phages, but none of them have been well defined in the taxonomy, this is something you would use to say, okay, these ones are all very similar at the nucleotide level, more than 70%. We've chosen a marker gene for phylogeny. We can see that this clade is well supported. We now say that this is a new genus. And that's what I would be happy with in a taxonomy proposal that just proposes a new genus. I think that is about the end of what we wanted to show you. So we've run through the whole tree building from selecting which sequences to multiple sequence alignment. We used the program MAFT. We checked the multiple sequence alignment twice, once before we trimmed, once after we trimmed, and then we did the tree building with IQ-tree. We let IQ-tree decide for us what the best substitution model is. If you want more information, I linked the Wikipedia page on substitution models for you. We trusted that it was correct. And then we visualized the tree with my favorite phylogenetic tree editor, ITOL.